Namaste, I am JV'N. I am Dr. ANUPAMA GOYAL. I am working as Associate Professor in Botany in Jyoti Vidyapeeth Women's University, Jaipur. Today I will deliver my lecture on RHYNIA, which is studied by the students of graduate as well as postgraduate students of various universities. Also, RHYNIA is a fossil plant. So students, today I will like to talk about RHYNIA, which is a fossil plant, and even some fossil plants are lepidodendron calamites. So first of all, I talk about the systematic position of RHYNIA. Well, it comes under division Pteridophyta, and the class is Xylophytopsida. Order of RHYNIA is Xylophytales, and the family is RHYNIaceae, and the genus we are talking about RHYNIA. So this RHYNIA is a fossil pteridophyte. It is a fossil pteridophytes, and now I talk about the features of Xylophytopsida. So Xylophytopsida is an assemblage of the simplest and earliest land plants. Their rootless sporophyte was differentiated into rhizome and slender aerial branches that were more or less dichotomously branched. The aerial axes were either naked or had small spirally arranged appendages. Thick-walled homosporous sporangia were terminal or lateral on the aerial shoots. The vasculature was protostelic. And now we continue. Xylophytopsida. The first member of this group was discovered by Sir William Dawson as early as 1859 from the rocks of the early Devonian period, and it was called Xylophyton due to its naked body. So students, what do you understand by Xylophyton? In Xylophyton, it is made up of two words. Xylos means naked and phyton means plant. The discovery of this simple plant did not fit into the preconceived notion of the land plants. Ramers placed the simplest fossil plants in the subdivision Xylophytopsida. It included only a single order, Xylophytales, and the following four families. So the four families are: first is RHYNIaceae, second family is Zosterophyllaceae, third family is Xylophytaceae, and the fourth family is Astroxylaceae. So we will talk about the family RHYNIaceae, in which RHYNIA has been placed. Now see, we are talking about the family RHYNIaceae. Well, the sporophytic plant body in RHYNIaceae was rootless. It had no roots, and it was differentiated into a cylindrical or lobed rhizome, and it was dichotomously branched, and it had erect aerial shoots. The rhizome bore rhizoids in tufts. The aerial axis had appendages, means true leaves were absent. So in the family RHYNIaceae, true leaves were lacking. Sporangia were borne at the tips of the branches, either singly or in groups, and when the sporangia are borne in groups, they are called as synangia. The sporangial wall was several layers thick. RHYNIA, Horneophyton, Cooksonia are some common members of the family Rhiniaceae. So the students, the question arises, name the members of the family Rhiniaceae. One of the members is RHYNIA. Other species, other genera or members are Horneophyton and Cooksonia. Now we talk about the distribution and habit of RHYNIA. The genus RHYNIA was named after village Rhynie in Aberdeenshire district of northern Scotland, where the first fossils of the plants were discovered. Two species are described from the red sandstone beds of Middle Devonian age by Kiston and Lang. In 1917, some 380 million years ago, RHYNIA and other plants grew in this locality. In marshy environment, there is evidence that these plants were growing in peaty habitats. They were growing in peaty habitats near the volcanoes where the atmosphere contained sulfurous vapors and the soil was saturated with acid water from the hot springs. The two known species of RHYNIA are RHYNIA major and RHYNIA vogani. The petrified remains of these plants are found, and on the basis of reconstruction, the form and structure of these plants have been described. Now I talk about the structure of RHYNIA. So first of all, I talk about the external feature of RHYNIA. The sporophyte plant body of RHYNIA was simple and consisted of a slender dichotomously branched rhizome bearing erect dichotomously branched aerial shoots or aerial stems. The aerial branches of RHYNIA major were about 50 cm in height and about 1.5 to 6 millimeter in diameter. The stems of RHYNIA vogani attained a height of about 20 centimeter and were only 1 to 3 millimeter in diameter. There were no roots, but from the rhizome grew numerous rhizoids. These stems were naked and had no leaves. The aerial branches seem to have terminated finally into sporangia. In RHYNIA vogani, hemispherical protuberances were present which might have arisen from the lower part of the aerial stems or from the rhizome. The vascular bundles of these adventitious branches were not connected with those of the main stems. It suggests that they were capable of growing into new plants if detached from the new axis, and they served as a means of vegetative propagation. So students, here you can see the diagram of RHYNIA major. Here the diagram A shows the sporophyte of, it shows the external structure of RHYNIA major, and B plant you see RHYNIA guani vogani. So as you see in the diagram, the plant or the fossil plant of RHYNIA has aerial shoots, and at the tip there is the sporangium, and the rhizomes and rhizoids are also present in RHYNIA plant. Now I talk about the internal structure of RHYNIA. Well, the internal organization of the rhizome as well as of the aerial shoot was very simple, and the following reasons were found. First of all, epidermis, then cortex. Now I talk about the epidermis. It was the outermost single layered envelope covered by a thick layer of cuticle. The epidermis of the aerial shoot was interrupted by stomata. Each stoma consists of two guard cells surrounded by many subsidiary cells. And now the cortex, well, the epidermis was followed by a well organized and broad cortex, which was differentiated into an outer and an inner region. The outer cortex consisted of 1 to 4 layers of compactly arranged polygonal parenchymatous cells without any intercellular spaces. This region represents hypodermis. Now the inner cortex was composed of spherical and parenchymatous cells with large intercellular spaces which maintained continuity with the outer atmosphere through the stomata present in the epidermis. This indicates that the inner cortex was the chief photosynthetic region in the cortex of some aerial shoots. Intercellular and intracellular fruiting bodies of fungal hyphae were also present. Now I talk about the central cylinder. Well, I am talking about the internal structure of RHYNIA. So a protostele was present in the central part of the axis as well as rhizome. In the stele, the xylem was surrounded by the phloem. The xylem was composed of only tracheids with annular or spiral thickenings. The phloem was represented by 4 to 4 layers of thin-walled elongated cells with oblique end walls. Some very minute sieve-like areas were present on the lateral walls of the phloem cells. The presence of sieve plate could not be ascertained. Endodermis and pericycle were also absent. Now I talk about the reproduction in RHYNIA. Well, the sporangia were cylindrical and borne singly on the tips of some aerial branches. Sporangia were large, oval or cylindrical structures with pointed ends. The sporangia had walls several cell layers thick, in which the cells of the outermost layer were thick walled and had a heavy cuticle. The middle layer was about 3 cells in thickness and composed of thin walled cells. The inner layer of the jacket composed of small rounded cells, and it functioned as tapetum. The sporangial cavity contained numerous spores of the same size. So the condition is homosporous. So the question arises, what do you understand by the term homosporous? Homosporous means the spores are of the same size and these spores have cutinized walls. The spores were apparently all alike and were arranged in tetrads. The presence of tetrads in some specimens suggests that they were formed by reduction division and that the plant bearing them represented sporophytic generation. The sporangium was without any specialized mechanism of dehiscence. Now I talk about the gametophyte, that is the spores in RHYNIA. The spores were cutinized and were borne in tetrads. The spores were 40 micron to 60 micron in diameter. Nothing is known about the gametophyte of RHYNIA because gametophyte of this fossil plant RHYNIA had never been discovered. So Lyon in 1957 reported some germinating spores from RHYNIA which show multicellular structures developing at the ends of the germ tubes that looked like that of RHYNIA major. These may represent the gametophytic generation, and Merker in 1959 has suggested that the underground parts of RHYNIA might possibly be the gametophytes. According to Pant in 1962, certain specimens described as RHYNIA guani vogani may be the gametophytes of RHYNIA major. However, no conclusive evidence of Archegonia or Anthradia is known to identify these gametophytes from the RHYNIA is known to identify these fossils as gametophytes. For example, or we can say Lionophyton, a recently described gametophyte from the RHYNIA does not seem to be related to RHYNIA. What I am telling? Lionophyton, a recently described gametophyte from the RHYNIA does not seem to be related to RHYNIA, but may be the gametophyte of other rhinophytes. Now see, this is the diagram of the section of the stem. This is the transverse section of the stem of RHYNIA guani vogani. So what it shows? It shows the outermost layer is the epidermis, as you see in the diagram, and below the epidermis lies the cortex, and cortex is differentiated into inner cortex and the outer cortex. Then you see the vascular cylinder or the central cylinder consisting of phloem and xylem. And now the last, you see this diagram shows the longitudinal section of the sporangium of RHYNIA. What you see in this diagram? The layer is tapetum and the jacket, and the spores, numerous spores can be seen in the sporangium, and these spores are found in tetrads. So here is the spore tetrads. Now I will talk in the next lecture about another fossil plant. Thank you for the sporangia were cylindrical and borne singly on the tips of some aerial branches. The sporangia were large, oval or cylindrical structures with pointed ends. The sporangia had walls, several cell layers thick, in which the cells of the outermost layer were thick walled and had a heavy cuticle. The middle layer was about 3 cells in thickness and composed of thin walled cells. The inner layer of the jacket composed of small rounded cells and functioned as tapetum. The sporangial cavity contained numerous spores of the same size, that is homosporous. Now students, what do you understand by the term homosporous? It means the spores are of the same size and the spores have cutinized walls. The spores were apparently all alike and these spores were arranged in tetrads. The presence of tetrads in some specimens suggests that they were formed by reduction division and that the plant bearing them represented sporophytic generation. The sporangium was without any specialized mechanism of dehiscence. Now I talk about the gametophyte, the spores. The spores were cutinized in RHYNIA and were borne in tetrads. The spores were about 40 microns to 65 microns in diameter. Nothing is known about the gametophyte of RHYNIA because gametophyte of this fossil plant RHYNIA has never been discovered. So Lyon in 1957 reported some germinating spores from RHYNIA chert which show multicellular structures developing at the ends of the germ tubes that looked like that of RHYNIA major. These may represent the gametophytic generation, and Merker in 1959 suggested that the underground parts of RHYNIA might possibly be the gametophyte. According to Pant in 1962, certain specimens described as RHYNIA guani may be evidence, may be the gametophytes of RHYNIA major. However, no conclusive evidence of Archegonia or Anthildia is known to identify these fossils as gametophytes. Well, Lionophyton, a recently described gametophyte from the RHYNIA chert, does not seem to be related to RHYNIA, but maybe the gametophyte of other rhinophytes. Now you see, this is the diagram of the transverse section of the stem of RHYNIA guani, and as you see in the diagram, it shows the outermost layer is the epidermis, and this epidermis is followed by cortex, and the cortex here is differentiated into outer cortex as well as the inner cortex, and the epidermis was interrupted by stomata, and now the central cylinder, as you see in the diagram, it consisted of phloem and xylem. Now you see the longitudinal section of sporangium of RHYNIA. Well, this atlas of rhinium sporangium shows the spores are arranged in tetrads, and we see the jacket layer and the tapetum. And now, thank you for hearing my lecture on RHYNIA with patience. Thank you so much.